Thank you very much for the invitation to speak today. My talk is going to be about the role that the PAX6 gene plays in brain development. So first of all, where is PAX6 expressed in the embryonic brain? On the left here is a three-dimensional image of an early human embryo, and this is from the Human Developmental Biology resource. And you can see this is curling towards you. You can see the head coming towards you. The eye is marked, the position of the heart, and the developing arm and the leg. Now, if we cut a section through the head, that's shown on the right-hand side of the slide, and you can see that the brown stain in this section is marking where PAX6 is expressed. And you can see that PAX6 is expressed in a layer of cells on both the left and the right side of the brain. And these are the structures that will form the left and the right hemispheres of the brain. And that layer of cells, each cell is a tiny dot. You can't probably really see it at this resolution. Each individual cell containing PAX6 is a small brown dot, and the cells that contain it are the dividing cells that will make the neurons of the cerebral cortex. So PAX6 is present in these important dividing cells in the cerebral cortex. So is it important for their development? What is the evidence for its importance? And there have been many studies of this over the years by many groups. The importance of the PAX6 gene in human brain development has been shown by groups such as that of James Lauderdale, Veronica Van Haneghan, and Sanjay Sisodia, and others. So, for example, this paper shown here in the top left shows a paper on the structural and functional consequences of PAX6 mutation in the brain. This is a recent review, and the evidence is now overwhelming for its importance. So today, what I'm going to do is to address this question, which is, why is PAX6 important in the developing brain? And I'm going to start out by giving what I think is the answer to this. I think that PAX6 is important because it controls the ability of developing cells to respond to their environment. And by that, I mean the immediate environment around the cells in the embryo. Now, all developing cells, in fact, all cells throughout life, are surrounded by a complicated mixture of molecules that can have profound effects on their development. And that complexity is summarized in this diagram. The details of this don't matter at all. It's just to give the overall message that for the cells that are embedded in this complex environment around them, there are many molecules here that can affect how they grow and how they develop. Now, in the most part, these molecules are important for the development of these cells, but it is possible that some of these molecules, their effects may be undesirable. For example, many of these molecules can have effects that are undesirable if they occur in the wrong cells or at the wrong times. So to put it crudely, we could imagine that the molecules around the cells have the ability to cause collateral damage. And so cells need to be able to control whether to respond or not to respond to the molecules around them. The ability of cells to respond or not to respond to their environment is termed competence in the developmental biology field. So cells, by controlling their competence, can control what they do in response to their environment. So to illustrate this, I'll talk briefly about one of PAX6's roles in eye development before moving on to the brain. So here's a nice diagram by Bell and colleagues showing how the lens is formed when the optic vesicle, which bulges out from the brain, approaches the surface of the embryo, the surface ectoderm. So the optic vesicle here is a dark blue structure. The approach of the optic vesicle induces the nearest part of the surface ectoderm, that's the skin of the embryo, to make lens. PAX6 is present quite widely in the surface ectoderm, and in fact in a much wider region than will form the lens. It makes the surface ectoderm competent to form the lens. It doesn't tell the cells of the surface ectoderm to make lens. Otherwise, they'd all become lens. Surface ectoderm cells might or they might not become lens. It depends exactly where the vesicle approaches, and that will vary slightly from individual to individual. Okay, so now I'm going to turn to the cerebral cortex. The picture in the middle here shows a tiny piece of cortex about 1 millimeter from top to bottom. Although it's so complex, it is in fact made up of only 2 major types of nerve cells, inhibitory and excitatory neurons. On the right is a blow-up of a few cortex cells, green for excitatory, green for go, red for inhibitory, red for stop. Excitatory neurons do what the name suggests. They transmit information by exciting or activating the cells they contact. Inhibitory neurons do the opposite. They moderate, they refine, they fine-tune the activity of the brain. They stabilize things. They prevent excessive activity. They increase the sensitivity of the brain circuitry. And normal brain function requires both of these 2 cell types to be made in the right numbers. Now, 25 years ago or so, a remarkable discovery was made in the field of developmental neuroscience, and this was that the excitatory and the inhibitory neurons of the brain are made in 2 different brain regions and only later combine in the cortex. So this diagram here shows a little bit of developing cortex in the middle, and here are the excitatory cells in green, and the blue cells underneath are the dividing cells, and these are actually the cells that in an earlier picture were in brown, and they express PAX6, and they will divide to produce the excitatory neurons of the brain. Now, what about the inhibitory neurons? The inhibitory neurons come from outside the cortex. They're immigrants into the cortex. They migrate into the cortex later on in a direction shown by these red arrows, the 3 red arrows here. So in summary then, we have 2 regions in the early embryonic brain. One contains cells that can, dividing cells that make PAX6, and it makes excitatory nerve cells. The other contains cells that don't contain PAX6, and it makes inhibitory nerve cells. So presence of PAX6 equates with excitatory neuron production. No PAX6 equates with inhibitory neuron production. Okay, so in our research, we set out to discover whether, and if so, why, PAX6 might be important in the cortex for the development of the excitatory neurons. So we deleted PAX6 in mice using some fancy genetic methods called conditional deletion to get rid of PAX6 from the cortex at the time when excitatory neurons were starting to be made. And these are pictures of thin sections cut through the embryonic mouse cortex. The red stain tells you where PAX6 is. I've been using various colors for PAX6. Sorry if that's a bit confusing. Brown in the early slides. Then I went to blue. Now I've gone to red. Forgive me for that. And the green marks the position of the cortex. And you can see that in the knockout on the right, the red stain has, for the large part, gone. So what happened when we did this, when we deleted PAX6 from the cortex? Now remember, PAX6 is in cells that make excitatory nerve cells and not in cells that make inhibitory cells. So what we found is that some, and only some, and I'm going to keep stressing this, only some cortical cells that no longer have PAX6 develop features of inhibitory neurons instead of excitatory neurons. And here are just 2 examples of many molecular features that we studied which are associated with inhibitory neurons. One of these features is the expression of a gene called GSX2 in red, and the other is the expression of a gene called GAD1, which is in purple here. And the 2 top pictures show the control cortex, and the bottom 2 show the effect of the knockout. And what you can see is that where there are very few or no red or purple cells in the cortex of the control, we now have these scattered red cells in the knockout and these scattered purple cells in the knockout. So I said that only some of these cells switched to become inhibitory. What about the ones that didn't switch and which didn't develop the features of inhibitory neurons? What we found is that these cells develop, as far as we can tell, like completely normal excitatory neurons. So although they've had PAX6 removed, they go ahead and make this cell type with no abnormalities that we've managed to detect. For example, in this experiment, we poked electrodes into one of the cells that looked normal, and you can see a trace of the electrical activity of one of these mutant cells, and each spike here in this picture on the right is a burst of activity called an action potential. And this is entirely normal and exactly how excitatory neurons behave. So, to summarize so far then, loss of PAX6 causes some cells to switch what they become. In other words, they change their fates. And I emphasize again, some cells do this. PAX6 is not required for other cells to develop normally. So our next question was, why does this happen? Why do some cells change and some don't? And what does it tell us about what PAX6 does? So one possibility was that the loss of PAX6 had altered the competence, remember that word from earlier, or the ability to respond, or the sensitivity, if you like, of cortical cells, making them more likely to respond in an abnormal way to molecules in their environment. In this diagram of 3 cells, the one at the top contains PAX6, that's a normal situation. And our idea was that this might protect it against the molecules that might induce the wrong type of nerve cell in their environment. And that's these green arrows. And you can see I've drawn them in different sizes here at the top, some small, some big, because these cells might be exposed to higher or lower levels of these molecules around them. So these are potentially inappropriate signals that these cells might be receiving, but this normal cell is protected against them by having PAX6. Now let's say that we take PAX6 away, and that's the second and the third of these cells, these blue circles. This on its own is not enough to make them develop into the wrong cell type. Perhaps only those that were exposed to high levels of this molecule in their environment took the wrong course, and that's the example at the bottom. So if there was a high level of this molecule, which the cells normally would be protected against, then without PAX6, they may go down the wrong route and become inhibitory neurons. Whereas if a cell happened to only experience or be exposed to a lower level of this molecule, then it may develop normally as an excitatory neuron, as shown in the middle example. So this could explain why some cells change and others don't when we remove PAX6. Okay, so what might this molecule be, this molecule that has the potential to derail them from their normal course of development? So one molecule that's capable of inducing inhibitory neuron production that's present widely in the brain is called sonic hedgehog, or SHH for short. And so we decided to test our theory that PAX6 deletion altered the sensitivity of cortical cells to SHH. And that's what's shown in this next slide. We took cells from the brain and gave them increasing concentrations of sonic hedgehog, and that increasing concentration is along the bottom of this graph. And we measured how many cells changed to become inhibitory neurons. The graph shows that for given concentrations of sonic hedgehog, more knockout cells became into neurons. So you see the solid line, the one without PAX6, is rising more rapidly than the one with PAX6. So if you pick out any sonic hedgehog concentration, for example, 10 nanomolar, then you'll find that more cells will switch if PAX6 is not present than will switch if PAX6 is present. In other words, if you just go up from the number 10 into the graph, you can see that. So control cells also respond, but not as well. And so we can say that the sensitivity to Sonic Hedgehog, or the likelihood, if you like, that a cell will switch to the wrong fate is increased when PAX6 isn't there. So let me sum up, and this is one of my students put it succinctly, and I quite like this term. PAX6 could be seen as a defender of the fates. It protects cortical cells against potential collateral deleterious effects, potential collateral damage from morphogens around them. Now, Sonic Hedgehog is very important, but we don't want it to operate in the wrong place at the wrong time. So, in conclusion, I think that PAX6 decreases the likelihood that brain cells will develop abnormally. It has a protective function. Now, the fact that this is a likelihood and not a certainty means that the precise effects of PAX6 removal will probably vary from individual to individual. And also remember, I've been talking about the extreme case of PAX6 removal. If levels of PAX6 were to be reduced, for example, by effects on only one of the two PAX6 genes that we carry, then there might be even more variability in outcomes from individual to individual. And one final speculation, if PAX6 protects developing cells from the risk factors that surround them, and if we could work out more about what those risk factors were and how they operate, we might be able to intervene in cases of PAX6 gene mutation in such a way as to reduce the effect of those risk factors. In other words, we might be able to help PAX6 carry out what might be its most essential function, to protect cells in the developing brain. So finally, I'd just like to thank all of those who worked with me on this project and the funders. Thank you.